always a treat to return to Berkeley. What I'm going to tell you about in the next 40 minutes or so is work that actually began here about 25 years ago. So bear with me, there's going to be a lot of history here. What I want to do is show you how basic science can really lead to insights into development of new therapeutics that are, as I hope to convince you, offer us the chance of using the word cure in the same sentence as cancer for the first time in a major way. Talk about some of the problems that we're facing and extending this to outside melanoma and tumors like that, and some of the solutions that we found that we're trying to find for other types of cancer. And there's my disclosures. Anyway, so as I said, this work really began here with studies, originally collaborative studies with David showing that CD28 was a necessary second signal for T-cell activation. But the real seminal findings that led to the development of this checkpoint blockade for treating cancer came from Max Krummel, who was a graduate student in my lab and is now a full professor at UCSF. First tumor experiments were done by Dana Leach. Sergio Quesada was a very talented postdoc that joined me when I was at Memorial Sloan Kettering, as was Mike Curran. I'll be talking about some of their work. Some Xiaozhou Fan, who was a graduate student with me at Sloan Kettering, and longtime collaboration with Alan Corman, who was here working at Bristol Myers Squibb at Medarex originally, and was really made the critical link that enabled us to get this therapy and developed by biotech after several years of frustration, and because it was too weird, as I'll tell you a little bit about it. My collaborator and partner in running the Immunotherapy Platform at MD Anderson, where we're really studying what goes on in patients. So why would you want to do immunotherapy for cancer? And I don't think I need to elaborate on that too much for this group, but basically there's three things that immunotherapy work offers that no other modality of cancer therapy works. The first is specificity, and I think everybody knows that T cells recognize peptides in the context of MHC molecules. But that's not really what I'm talking about. What I'm talking about, there's increasing data showing that the immune system in cancer is focused on foreign antigens, neoantigens that are generated by the mutational process, genomic instability that's inherent in the cancer problem and the causation of cancer. So the immune system is really focused not on any one particular driver mutation, but just on the mutations in general that accompany the process of carcinogenesis. And of course, you'll hear more about this from Rafi. Once you have T cells, you've got them basically for the rest of your life. If the tumor comes back, the immune system will be reactivated to attack them. And finally, adaptability. After the identification of the antigen receptor and cloning of the genes, Mark Davis calculated that the alpha-beta recombination machinery can generate 10 to the 15th power different TCRs. And of course, that's billions of more cells than you have in your body. But everybody has about 100 million different ones circulating around that have the ability to recognize virtually anything that nature throws at it, including these mutations that really hamper, that happen constantly in tumor cells that allow tumors to escape treatment with conventional agents that just attack one mutation. So the immune system can change, and since it's a living organism, and adapt to the heterogeneity and the adaptability of cancer, which can develop mutations in 23,000 genes. I think the immune system is the one thing that is a fair match for that. And so where are we now? I'll just start with this to set the stage for why there's a lot of interest in this. So ipilimumab, which was David talked about a little bit, was the first drug. It was approved by the FDA in 2011 for the treatment of metastatic melanoma. And so it was a few years before there were other approvals. Two different PD-1 antibodies, you'll hear a lot about one of them from our Melman, were approved for melanoma in 2014. Last year there were 5 approvals of these basic drugs and combinations in lung and in renal cell carcinoma. The speed at which this field is advancing is illustrated by the rapidity with which nivolumab, the BMS PD-1 antibody, was approved for metastatic renal cell carcinoma. The first trial was completed and reported in September of last year, and it was approved by the FDA in November. So things are really accelerating in this space. And this year we're going to see at least 5 and probably more approvals than that. So this field is advancing at a geometric rate. And I think you'll hear from Ira, and I want to also make the point that it's going so fast that the science is beginning to fall behind what we understand here. And I want to illustrate this towards the end of the talk. Anyway, where did this come from? Well, it really was a paradigm shift in cancer therapy, because you'll see this therapy does not target tumor cells. And so when I started trying to get companies interested, they said, well, how can you treat cancer by ignoring the cancer cells? Well, because we really focus on the immune system, and it also does not involve turning on anything. People say, we harnessed the immune system to treat cancer. We didn't. We unleashed it by blocking inhibitory signals that allow it to attack things that were already there. So it's not really harnessing, it's just letting it do what it would do. And the way we got there wasn't through the war on cancer or through studying the genome or sequencing anything in cancer cells. It was by fundamental science and understanding just the real basics of how T cells are regulated. It all came out of that. And anyway, I think you'll see I want to make a real push for fundamental science. We need to keep doing that. We're going to really understand how to extend this. And so this is really the central feature that we're going to be talking about here for most of the talk. It was realized after the antigen receptor was identified, became apparent really quickly that signal was not sufficient to activate T cells or additional co-stimulatory signals that were provided by antigen-presenting cells, by dendritic cells in particular, for which Ralph Steiner won the Nobel Prize several years ago. But at Berkeley, the question was really generated by Ron Schwartz at the NIH, these co-stimulatory signals. And we showed here in my lab in the early 90s that CD28 was sufficient and necessary to provide a second signal in addition to the antigen receptor to get a naive T cell activated. And I won't go into detail through it, but it turned out there was a molecule called CTLA-4 that had already been cloned. It had an unknown function. It was highly homologous to CD28. The only thing that was known about it, after we cloned CD28, we came across it, as did others. The only thing that was known about it was it wasn't in resting T cells and was turned on after activation. And after a lot of work, Jeff Bluestone's lab and mine, Max Krummel, as I mentioned before, showed it's really an inhibitory molecule. It's turned on at activation. So there's two important things that happen that weren't really recognized before this. One of them is that when you get the TCR signal and CD28, you turn on all this stuff, which drives a T cell towards rapid proliferation. They have to go from a few hundred to tens of thousands of cells, if not hundreds of thousands of cells, in a matter of 4 or 5 days to deal with a virus infection or whatever. So you turn on this stuff rapidly. But when you do that, you also turn on CTLA-4, which accumulates, binds to the same ligands, and eventually stops an immune response. You have to have that to take care of that rampant proliferation of T cells, which will kill you if you don't stop it. And that was shown by Cynthia Chambers in my lab, the late Cynthia Chambers and Tack Mack and Arlene Sharp and others. If you knock out CTLA-4, mice die when they're about 3 weeks old because they just fill up with T cells. They basically turn into giant lymph nodes. And so that's kind of how the system works. But in cancer, it's more complicated because solid tumor cells, at least, did not have co-stimulatory activity. That was shown by Ron Schwartz and Mark Jenkins. So we did some experiments before CTLA-4 was discovered where we put the B7 ligands, the co-stimulatory ligands, in tumor cells, and they would immediately get rejected. The mice would be immune. So they've got antigens, but they're invisible to the immune system because they can't co-stimulate until they die. Maybe they get too big, cut off the food supply or something. But anyway, they die, and then the dendritic cells come and phagocytize the dying tumor bits, and through the STING pathway that David mentioned that Russell Brand studies, it gives a signal to the innate immune system to make type 1 interferons and upregulate the antigen processing machinery, reprocesses antigens from the tumor cell, displays them in the context of the co-stimulatory molecules, and that turns on the response, but also turns on CTLA-4. And so the notion that we had is the tumor's gotten a head start. And so it's invisible for so much time. By the time it starts, if you turn on the off program, if your immune system doesn't eliminate all the tumor cells before that off program reaches a level where it shuts it down, then the immune system will not take the tumors out. So we had the idea of just disabling the brakes by putting CTLA-4 antibodies in the thing, taking the CTLA-4 out, and just allow co-stimulation and activation to go on as long as we want. And we can control it by just stopping giving the antibody. So we thought it would work as a monotherapy in necrotic cell death, or you could combine it with vaccines, or combine it with virtually anything that kills tumor cells and starts this cross-priming activity to really provide conventional drugs, perhaps, with memory, with adaptability, and all the things that the immune response provides for it. So it was a fun idea, but the question was, would it work? And this is one of the first experiments we did. This is a transplantable tumor growing on the backs of mice. You can see it grows you have to euthanize the mice. If you block CD28, it grows faster, because showing that there is a nascent immune response, but that requires CD28 cross-priming, but it's not sufficient to eliminate the tumor. But when you inject anti-CTLA-4, the tumor grows for a while and then goes away, and the mice are permanently immune. And so when we got this result, I was really startled because of all the things that are going on when a host immune system contacts tumors. Covering up this one molecule was sufficient to get rejection and long-lived immunity. But it does take some time. The tumors grew before they regressed. And so this had important issues that were in the clinical development that I'll come to later. Anyway, it doesn't always work. The B16 melanoma had been thought to be essentially non-immunogenic. CTLA-4 blockade did nothing to that tumor at all. Glenn Janoff had shown about the time we were doing this work that he could prophylactically vaccinate against B16 by putting the GM-CSF cytokine gene into the melanoma cells, irradiating them, and then injecting them, and they die in a big cloud of GM-CSF, which helps dendritic cells come and mature and all that. But therapeutically, it didn't work at all. But when we combine these two things, that to enhance cross-priming, this to sustain cross-priming, 0 + 0 resulted in essentially complete eradication of the tumor. So, a truly synergistic results where neither thing alone had any activity at all. You put them together, and the tumor gets rejected. And so just to look at what goes on, you can see this is an untreated tumor in mice. You can see the vasculature in red, the odd T cell out in the tumor. If you look at the kind of T cells that are there are a lot of CD4s. Most of them have purple nuclei. That's FOXP3, a transcriptional factor that turns them into regulatory cells that make TGF-beta and IL-10. So that's a very suppressive environment. But when you give anti-CTLA-4 in the vaccine, you can see the vasculature gets activated, expresses the integrins ICAM and VCAM, not shown here. And that allows T cells that are going through the vasculature in the tumor to extravasate, which you can see occurring here. But now if you look in there and you see the T cells, there's obviously a lot more T cells. There's still a lot of CD4s, but most of them do not have FOXP3 now. They make TNF alpha and gamma interferon and eliminate the tumors. There's also CD8s, the big granzyme. So anyway, after, again, I won't go through all the stories, but 2 or 3 years of frustration, we finally teamed up with Metarex, due to the efforts of Alan Corman, and produced a fully human antibody that was named MDX-010. It was a lot of fun in the early days because we'd talk about the 010 project, the MDX program. But then when it got to the FDA, they named it ipilimumab, which is a horrible name, very disappointing name. And so I told Metarex that, especially because the work came out of Berkeley and I was at Berkeley, that they just ought to put an H in front of it and call it ipilimumab in honor of its Berkeley origins, but they wouldn't do it. So I guess they decided ipilimumab didn't have enough gravitas for a cancer drug. But anyway, later, Metarex teamed up with Bristol Myers Squibb. There have been over 50,000 patients treated. There have been, as we would have predicted, objective responses in many tumor types. There are adverse events. They're not autoimmunity. They're itises, a lot of inflammatory responses that can be dealt with by temporary treatment with steroids, which have been withdrawn and they don't come back, so it's not autoimmunity, and they don't seem to interfere with the anti-tumor effect. So this is one of my favorite slides, and Dan was mentioning this earlier. This is probably the first, anyway, this woman was in the phase 1 trial. She had failed every other therapy. And you can see this for melanoma. You can see this lung mass here and this pleural effusion, including high-dose IL-2, a dendritic vaccine, and other stuff. And Tony Rebus at UCLA was treating her and said, he had this drug, first in man. Would you do it? There may be some risk. She said, yeah, she just wanted to live a few months, just long enough to see her kids graduate from high school. I visited there 10 years later in 2011, and she had just had her 10-year follow-up, and she was tumor-free after a decade with only 1 injection of the antibody. And she's still doing fine. I contact her every now and then. Anyway, she's 15 years out now, again, after a single treatment. So just cut to the chase. This was a randomized phase 3 trial in metastatic melanoma. Has 3 arms, the CTLA-4 only arm here. This is with the peptide vaccine. We can ignore that for now. And then this is the placebo. The median survival after diagnosis with metastatic melanoma at the time was 11 months. And so there was no standard of care. So it was ethical in this case to use a placebo control for some of the patients. I should point out that when this trial started, the endpoint was going to be just progression-free survival. Despite the fact in our mice, they almost always progressed before they regressed. And so there were 2 or 3 failed phase 3 trials that had just progression-free survival as an endpoint. So Medarex, Bristol Myers Squibb and Medarex decided to change the endpoint in the middle of the trial from progression-free survival to overall survival. So the trial took over 4 years. But what you can see is there's an increase in the median of about 4 months with ipilimumab. So that would have been sufficient for approval. But the cool thing is that it flattens out about 2.5 to 3 years and stayed flat for the remaining period of the trial, which means basically it's a tail on the curve. So people are not dying after 2.5 years. And so that was approved by the FDA in 2011 and is now a standard of care for metastatic melanoma. But there was anyway, there was a 10-year follow-up of almost 5,000 patients that was released in 2014. And you can see that that inflection point occurs about here. 22% of the patients were alive 10 years after a single round of treatment, which is 4 doses at 3-week intervals. So about, as David mentioned, 22% of the people I think could be considered cured of metastatic melanoma. I've actually got one of these, I don't know why I'm screwing up. So anyway, that was CTLA-4. And so why did more patients respond? Well, one reason is because there are multiple checkpoints, and Rafi's going to talk a lot about PD-1, but this is a molecule that was cloned actually by Tosco Hanjo in the early 90s. But in about 2001, Gordon Freeman and Arlene Sharp showed that it was a checkpoint. Actually, it wasn't a molecule involved in negative selection as had been previously thought. It has 2 ligands like CTLA-4 that are on antigen-presenting cells, but it differs in that it can be induced by gamma interferon on tumor cells themselves, and it seems to interfere with T-cell receptor signaling, not co-stimulation. So just to in this part, in early phase 1 trials, it, like CTLA-4, showed activity in several kinds of cancer, including melanoma, with objective response rates about 28%, non-small cell lung cancer and renal cancer. There were no responders in colorectal cancer, unlike IPI, which had activity there. But now it's been shown that there is a subset of patients with colorectal cancer that have microsatellite instability defects in DNA repair, and so have a lot of mutations, a lot of new peptides, and so that subset responds at a very good rate. There are also no responses in castrate-resistant prostate cancer, and I'll show you why that is towards the end of the talk. Anyway, this is a typical survival curve for melanoma patients treated with PD-1 antibodies. It's not clear how good a tail there is on this, and with time we'll see how well that works. But anyway, it was shown that patients that don't respond to CTLA-4 respond to PD-1 and vice versa. And so, how do you get the response rate up? Well, the obvious thing is to combine them. So Mike Curran in my lab in mouse models started combining the different antibodies to CTLA-4, PD-1, and PD-L1, could show that there were additive effects at least. And so this and some work from Alan Corman led to a trial in which CTLA-4 and PD-1 were given together. And this is a concurrent delivery of them. What you can see here is that 2/3 of the patient, the spider plot is tracking the volume of tumors in individual patients. 2/3 of the patients showed some evidence of tumor shrinkage, and 50% of them had what would be called objective responses. This is the complete disappearance of the tumor or disappearance of at least 50% of it. And this is a waterfall plot, it's called here, that just at an index point shows the change in different patients. You can see the majority of the patients responded. Half of the ones had 50% or more shrinkage. Half of those that had objective responses had 80% or more tumor shrinkage. And so this was pretty amazing, a 50% response rate. But this raised hope in a lot of us about what the survival benefit would be, because for reasons that have to do with the fact that CTLA-4 can operate slowly, the objective response rate in the ipilimumab trial was 9%, but the survival benefit was 22%, as I said. So this is all objective response rate, and so many of us thought that 50% rate is going to underestimate the survival benefit that patients would have, because it grossly underestimates the survival benefit in ipilimumab-treated patients. And so this is a follow-up just at 2 years after a phase 1, and I have to say that this is not a randomized trial, it's a small number, it was in a phase 1, but at 2 years, 88% of the patients in that trial were still alive. So I told you that, again, the mean survival at that time, median survival was 11 months. And so if these patients make it to 3.5, they might be expected to go for 10. So we don't know yet, but this combination was approved by the FDA last year, and so it's now a standard of care, and we'll know because there are going to be large numbers of people treated with this. But it shows what you can do by doing combinations and really having just really changed the situation in melanoma. So these are the two main pathways that are now in play. They're similar, they're both checkpoints, but they got certain differences. CTLA-4, as I said, is hardwired in there. It's turned on when you activate a cell. PD-1 seems to be induced resistance by gamma interferon, inducing expression of PD-L1 on the tumor cells. This targets CD28. This is thought to target TCR signaling. I may talk about some data that suggests that it may not be correct. There's pretty good data suggesting that CTLA-4 blockade brings new clones of T cells into the response, whereas PD-1 doesn't seem to. It just seems to expand the clones that are already there. As I'll show you, CTLA-4 can move T cells into a tumor. It looks like PD-1 really can't. It can increase the numbers that are in there, but it does not seem to drive them in. And disease recurrence after response to IPI is very rare, but there is significant relapse of patients with PD-1 in melanoma and lung cancer after they've responded. So again, fundamental difference between these two things, and it's really critical that we figure out, why that is. So anyway, we're at a point now where we've got a good situation. We've got a grip on it. We can cure certain patients. But again, I keep harping on this, but we need to really understand the mechanisms that are involved, see if we can have predictive prognostic or pharmacodynamic markers. If we understand mechanisms, we can in a rational way start to put these things together and finally try to find new molecules to improve efficacy. So when I moved to MD Anderson, we started what's called Immunotherapy Platform. And I'm sure everybody's familiar with the phase 1, 2, 3 pattern, safety, then efficacy, and then comparison to standard of care, which is really pretty much obsolete now for many of these drugs because you see responses, you see efficacy in phase 1. In fact, the first PD-1 antibody approved for melanoma was just a giant phase 1. It never went to phase 2 or phase 3 for approval. So we've changed this a little bit, and we're more interested in the clinical endpoints and mechanistic endpoints. So we're specializing in developing small pre-surgical trials where we can get the whole organ to study what's going on, biomarker analysis and mechanistic insights. And I'd like to quickly just show an example of this. It was actually done by Pam Sharma at Anderson. So she was studying bladder cancer, localized bladder cancer, which is dealt with by cystectomy. It's removal of the bladder and a reconstruction of the bladder. When that's done, they also take the prostate out. So this is a 12-person trial that she did, 6 men and 6 women. Half the men had prostate cancer in addition to bladder cancer. And so she got the entire organ with all the tumor except what the pathologist needed for diagnosis. But that gave enough T cells to do, biopsies of that to get enough T cells to expand or do flow cytometry, histology, functional studies, expression arrays, whatever. Really is quite a powerful thing. And so this just shows what happened in bladder cancer, just doing expression arrays with the untreated patients and patients that have been consented to get 2 doses of anti-CTLA-4 before they went to the surgery. It was going to be curative anyway. But these patients were courageous in undergoing a procedure that was probably not going to do them any good, but would provide information on what's going on. And so the number one change was, these are all, as you'll immediately see, these are all T cell signatures. The most of highest significance was the ICOS pathway, and this was really unsuspected. We had no idea this was going to happen for reasons that I'll tell you in a minute, but Pam looked at it, and so the expression arrays were confirmed by looking at by flow cytometry. So there's about 13% of the CD4 cells in non-malignant, untreated tissues, bladder tissues, In the tumor, it's about the same. But after they get anti-CTLA-4, it goes up. In this patient, 3-fold. Sometimes it goes up 8-fold or 10-fold. And this also happens in the blood. This is the first marker that anybody had found that changed in the blood significantly and could tell you when the antibody had worked. Before then, people were just trying to measure whatever they could think of to measure in blood and do enough patients to see if there's a statistical significance in treated versus non-treated. And it was all blinded, and none of that stuff really held up. But what went on in the tumor guided what to look for in the blood. So why were we surprised? Well, if you ask an immunologist what ICOS positive cells are, there's really two types. One are follicular T helper cells. These are in germinal centers, and they make type 2 interferons, IL-4, IL-10, and stuff that help B cells class switch their immunoglobulins. The other cells are a subset of Tregs, where ICOS regulates IL-10 production by the cells. Neither of those, of course, would do you any good in a tumor, and yet that was the most obvious change of what happens. ICOS, just if you don't know, is a close relative of CD28 and CTLA-4. It's on the same little bit of DNA. It has its own unique ligand, ICOS ligand, but it had never been reported in tumor situations before. So what Pam did was type the tumors from the patients. 3 of them expressed RNA from a cancer testis antigen called NY-ESO-1 that's epigenetically regulated. So she sorted the ICOS high cells and tested them against peptide pulsed APCs from the patients. You can see that all 3 of them, the CD4 high CD4s made, ICOS high CD4s made gamma interferon and also TNF alpha in response to tumor antigen. Virtually all of the anti-tumor activity in the CD4 population were in the ICOS positive population. This patient already had them before therapy. So this was very interesting and raised the possibility that this may be functionally important. So I raised that they may be important. So one of the first things we did was while I was still in New York, was to thaw about 140 patient specimens pre-treatment and then after the completion of therapy. And they were scored and put into 2 bins, those in which the ICOS frequency went up at least 2-fold and stayed there for the whole 3 months of therapy, or didn't go up or just went up transiently and came back down. So those with transient expression, and when we looked at their survival, they had a median survival of about 8 months. The ones that when it went up and stayed up had a median survival of over 20 months. And so that suggested that it might be important in the therapeutic effect. But of course, these patients at the time that this was going on were all heavily pretreated with chemotherapy. So an equally possible explanation for these patients' failure to respond is that their immune system were just too beat up to do anything. And one of the lessons that comes out of these sort of studies is you really can't test hypotheses in clinical trials. All you can do is generate hypotheses, and you really need to take it back to mice to figure out what's going on. So we went back to the mice in that same tumor model that I showed you earlier, which cured in this experiment about 85% of the mice. If you do it in ICOS or ICOS ligand knockout mice, you lose more than half of the efficacy. So this is important in the clinical benefit from anti-CTLA-4, and we've refined this now. There's a PI3 kinase binding site on the cytoplasmic tail of ICOS, and if you change the tyrosine in that to a phenylalanine where it can't bind PI3 kinase, you lose the efficacy too. So it signals through the AKT pathway, and that's the mechanism by which it enhances it. And so this raised another hypothesis that maybe we could target ICOS to increase the efficacy of CTLA-4. So what we did in my lab was to make something like the GVAX that Ben Dranoff had developed, except we put in the ICOS ligand into the tumor cells, irradiate or not, and gave the wild type irradiated cells with the ICOS ligand vaccine to mice with or without anti-CTLA-4, and then looking at what happened. These are all the monotherapies here, and in this version of the experiment, you can see here, this is the anti-CTLA-4 plus the wild type vaccine, about 15% or so efficacy that goes up to close to 80% by just having the ICOS ligand in the vaccine. And it's due to the signaling, because if the recipients don't have ICOS, you lose the benefit. So again, we can target CTLA-4, so target ICOS to improve it. What happens is really pretty remarkable. The density of CD8s increases about 10-fold, but the cells that could degranulate in response to tumor antigen increased 3-fold. So there's overall about a 30-fold increase in effective CD8 killers. In CD4, there's only about a 3-fold increase in the number of cells in the tumor, but there's a 30-fold increase in the number of them that are polyfunctional, making both gamma interferon and TNF-alpha. So we think that's really what's responsible for the increased efficacy of this treatment. So we started a small company that's made an agonistic antibody to CTLA-4 that should be going into the clinic at the end of the year. So just to wrap up, we've got, what I didn't mention was that that elevation of ICOS is a pharmacodynamic marker. It's been shown to really tell you who's the CTLA-4 has actually hit the target, and it doesn't tell you necessarily who's going to respond, but it tells you who's not going to respond, because if that doesn't go up, the patient should be given something else pretty much immediately. So anyway, this is one of the most commonly shown slides in immunotherapy these days, but this just shows that different tumors have different mutational loads. And what's immediately apparent is that the tumors, melanoma and lung cancer, for which monotherapies work very well and have been approved, are really high in mutational load. Again, colorectal cancer, which is here, there's a subset. These guys are the ones that tend to respond to PD-1, those that have microsatellite instability. Kidney cancer also responds very well, even though it has a relatively low burden, but it's not quite sure. There's something special about kidney. But we've been kind of interested in prostate cancer and other tumors, pancreatic cancer and glioblastoma, the tumors down here. And as I mentioned earlier, PD-1 didn't work at all against prostate cancer. CTLA-4 works about 8% of the time, but it failed in a phase 3 trial. So it's really not considered immunogenic target that could be used with checkpoint blockade. Bristol Myers Squibb stopped their whole prostate cancer program after the failure of those trials. But when we started thinking about this, and this is from a review article that Pam and I wrote, but there's a lot of literature out there supporting this, that the tumors at the high end of the mutational load tend to be inflamed or hot or immunogenic, whatever you want to call it. They've already got a lot of T cells in them, so all you need to do is take the brakes off and you can get a response. But tumors at the lower side of that tend to be not so heavily infiltrated, and so there's nothing to operate on. So you've got to do something to get the T cells in to really, be able to do anything. And so there's several ways of doing that. I'm just going to show you one. So this is patients that actually, this is pre-treatment immunohistochemistry, and then after treatment with anti-CTLA-4. And what you can see here is there's a huge increase in CD4s, CD8s, the ICOS positive cells, CD45RO, granzyme B, and a lot of macrophages come into them. Another way of looking at it is here. Now this is an untreated pre-treatment tumor, and this is the tumor here, and you can see there's not very much going on in there. There's some CD4s here and there. There are areas outside the tumor where there are a lot of T cells, but they're not inside the tumor. But after you give anti-CTLA-4, they go in. There's robust infiltration of CD8s, CDs, macrophages. But if you look at, I don't know if anybody can see this. I can't see it very well. But anyway, this is multiplex immunohistochemistry, so we stain with 6 colors at the same time. And this shows the CD8 cells. And when you reveal the PD-L1 staining after that, the CD8 cells have a lot of PD-L1 on them, as is shown. Well, this just shows it again. This is untreated here, and this is post-treatment CD4, and this is PD-L1. So there's not any PD-L1 on the CD4s. There's lots of PD-L1 on the CD8s, and there's lots of PD-L1 on the macrophages. And that's just shown here, and also on the tumor cells. That's what you see. So the reason that that CTLA-4 didn't work is because you get the T cells in, but then PD-L1 comes up and can inhibit through this other pathway. But I don't know if I'll talk about this or not, but this begins to call into question our understanding of the mechanism, because the common mechanism is, the understanding of the mechanism is what I showed you in the slide. The T cell gets near the tumor cell, makes gamma interferon, the tumor cell upregulates PD-L1. So the fact that it's also L1 is upregulated on macrophages and T cells themselves suggest additional mechanisms that nobody's really investigated yet, but it's not as simple as perhaps not as simple as the cartoons suggest. So anyway, we went back to BMS, showed him these data, said, well, PD-1 doesn't work because there's no T cells. CTLA-4 blockade doesn't work because there's PD-L1 all over the place, so just do them both at the same time. And so we're going to be opening a trial doing that in about 2 months. But it's more complicated than that even. There's another molecule called Vista that Randy Noel described that's another inhibitory molecule, and that also appears in cells, not so much on CD8s, but really on macrophages, really high levels in macrophages on the patients that have been treated with anti-CTLA-4. And the numerical data are shown here. The macrophages really are screaming with Vista. And so that may be another mechanism. If the PD-1 and CTLA-4 don't work, then we might have to add cocktails of Vista development, which we're helping a small biotech company develop at this point. Anyway, I'll just finish by saying everything that you could think of being done is being done in this. Sometimes not so rationally, other times with rational studies, but based things. But there's all this stuff that can be done. The combinations that exist now are so many that there's not even enough patients. So we've got to again get some understanding of the mechanisms that are involved, so we can not just pick one from column A and one from column B and another from column C and mix them up. But there's a lot of promise. And there's also, I mentioned Vista, but there are also other molecules, TIM3, LAG3, other molecules that are in development that are these multiple checkpoints. One of the things that we're discovering is there are many checkpoints that many of them probably won't be important. Many of them will be dominant. CTLA-4 and PD-1, I think, are probably the most important. It's given, at least in melanoma, it's given by the response rates, but there's a lot more to do. But the exciting thing is that until recently, the whole goal of cancer therapy was to move the median survival over a few months and declare victory and then move on. But what we know now from CTLA-4 treatment is you can cure a fraction of patients, 20% or so. And we know by the combination with PD-1, we're gonna raise that. This is not real data. This is just an aspirational goal there that we would like to have. But of course, it's possible now. So the goal shouldn't be this so much anymore. It shouldn't be moving the median. It should be raising the tail as much as we can and do this in many different kinds of cancer. But as I said, I think there's a lot of reason for optimism, and it's just a matter of continuing to do the science and understand how to put everything together to do this. So I'll end with that and hope I didn't go over too much. But anyway, thank you for your attention.
Time for some questions.
Let me get some water.
Maybe I can start if there's no takers yet. Yeah, so I'll feed you a little bit of a softball here, but I think it's actually a substantive question. To what extent do these checkpoint blockers or inhibitors just affect immediate effector activity versus inactivating the T cells in a more stable fashion?
I think you'll hear more about that from Rafi actually with PD-1. It can do both, but it seems that combinations of them to get the most exhausted, correct me if I'm wrong, T cells are the ones that have PD-1 and TIM3 and LAG3, all three of them together. Right. Is a marker of really, the most exhausted cells or whatever. Right. So PD-1 can immediately stop cytokine production.
Right.
Can you comment on the oncolytic virus in terms of, for example, TVEC combination with inhibitor blocker?
The oncolytic viruses, yeah.
Yeah, oncolytic virus with immunotherapy.
Yeah, we published a paper a couple of years ago using Newcastle disease virus, and it's very effective. It engages the innate immune system through. It's an RNA virus, so there's sensors for RNA in the cytoplasm of dendritic cells, of tumor cells. So what we found is you inject it into a tumor, you can, just with the virus, blow away the tumor that you inject. But if you you see some increase in a distant virus, a different tumor with T cells in there, but you don't get rejection of that tumor unless you give anti-CTLA-4, and then you can get all the tumors cured in most of the mice. So that combination is now under development. We also more recently have a paper submitted now where we put ICOS ligand into the virus, and that makes it work even better because it gives it that little ICOS stimulation. So.
There's one.
You mentioned in your talk that the survival rate basically has an additive effect when you're blocking CTLA4 and PD-L1. But you mentioned that for PD-L1 that the actual disease recurrence actually happens more often than CTLA4, and those seem contradictory effects to me. I was wondering if you understand.
Well, we don't really understand why there's relapse. The reason with CTLA4, the objective response rate. Objective responses are measured at a certain point in time, and usually it's 3 months or so. And so it's known now that I mentioned that the reason they changed the endpoint of that trial is it was known that a substantial number of patients still have tumor progression, at the time they're evaluated, so they don't count as objective responders. And that's why I said that because of that, two very large phase 3 trials failed. But that's the difference. And most of the patients get PD-1 respond quicker, probably because as David mentioned, it works at the level of effector function, whereas CTLA4 works at the level of priming, and so it may take longer. But why they're failing, we don't know. There's some evidence that there's problems with the gamma interferon signaling pathway. That's one mechanism at least, but it's something that's just now beginning to be studied.
Down here. She's going first.
Sorry. Can you comment on potential with combining also growth factors, the IL-15 or 2-based therapies?
Combining growth factors?
Yeah.
It's been tried. The combination of IL-2 plus anti-CTLA4 has been tried. There was a slight increase in efficacy, but it really wasn't worth the toxicity of the IL-2, so that's been discontinued.
I just want to pick up on something you may have mentioned just offhand. If it's so that we all expect that CTLA4 would act primarily at the level of priming. If that's,
I think that's correct.
Yeah, who knows? But we assume that that's correct.
Yeah.
If that's so, does that imply then that there's a continuous need for priming in patients in order to really mount and sustain
I think to get
response?
a sustained response, they do after a while have to be rejuvenated. At least that's what I think. I mean, I don't know how you prove that, but they have to go back and get another shot of anti-apoptotic factors and IL-2 production and all that. I don't know. Other immunologists may want to dispute that, but that's the model.
Any more?
Yeah, thanks for the talk. It was really good. You showed some evidence, and there's correlation of diseases that are most responsive being diseases that have the highest rates of neoepitopes or cancer mutations. But how much evidence is there that the disinhibited T cells, the preponderance of them anyway, are really recognizing mutated epitopes versus, say, wild-type epitopes that are in compartments that are not tolerized and just haven't been surveyed by the immune system before?
Yeah, well, I'd have to admit that there's no real hard data suggest. It's really difficult to tell which is going on because they're both going on, right? I mean, in melanoma there are definitely T cells being developed that recognize tyrosinated proteins, but it's really easy now with the new technology to get, to predict the epitopes generated by mutations and then demonstrate that they're there. So they're there and they can kill tumor cells. Steve Rosenberg has actually defined a neoantigen that was recognized by a CD4 cell and engineered T cells to express that receptor and has cured patients with it. But you can do the same thing if you put in a massive number of MAGE-specific T cells, for example. But I think that the main reason that people accept that is because to all the tissue-specific differentiation antigens, the high-affinity T cells are going to be taken out because of central tolerance and peripheral tolerance mechanism, which should not be, at least central tolerance should not be an issue for the neoantigens. And these cells have been proven to have much higher avidities, for example, for seeing the epitopes.
Thank you.
But there's no direct, we're trying to do that now in prostate cancer, actually. We've got 20 patients that we've sequenced, and we're looking for both the neoantigens and PAP and PSA specific T cells, stuff like that.
Okay, thank you, Jim.